Adaptation. The process by which natural selection makes an organism better suited to its environment. Changing existing features in order to best exploit existing sources. Or to disguise an organism from predators. Sometimes, natural selection can cause an organism to lose pre-existing features, much as these cavefish have lost their eyes after thousands of years in pitch darkness. But more intriguing are those cases in which natural selection has caused organisms to develop novel features that their ancestors lacked, such as this bombardier beetle's ability to douse its predators with caustic liquid. In order for natural selection to operate, each intermediate between the ancestral organism and the evolved organism must be slightly more fit than the previous intermediate. Ultimately, every new feature of an organism must spring from step-by-step refinements and adaptations of its ancestral features. The complex bacterial flagellum adapted from a needle pathogenic bacteria used to inject infectious proteins and toxins into other cells. While some express this belief that something as complex as the eye could have evolved, one may trace the evolution of an eye through many stages. From simple patches of photosensitive cells, to patches of photosensitive cells with cups, to pinhole camera style of eye without a true lens, to eyes with simple lenses, to eyes with complex focusable lenses. Each stage of this process co-opted and improved existing features into a larger structure. While it is possible to infer how evolution happened in the past through good detective work and an excellent knowledge of the organism's anatomy and physiology, laboratory work allows us to watch evolution and adaptation in action. In OEB 100, students are analyzing how bacteria have evolved over relatively short periods of evolutionary time, hundreds of bacterial generations, or about 2 months. The bacteria that we study have been coaxed into growing in hostile environments, those containing poisons or lacking their usual food. While usually sorely pressed in the short term, these bacteria soon evolved and thrive in their new conditions. These physiological changes are rooted in the changes in the bacterial chromosome. In our work, knowledge of bacterial physiology feeds and informs our study of adaptation. We may examine the frequency of mutations in metabolic pathways predicted to handle the bacteria's new environment, or we may look at loci in which we have observed many mutations in order to elucidate undiscovered portions of bacterial metabolism. We are also investigating new methods of laboratory evolution, such as forcing bacteria to climb gradients of formaldehyde in order to get access to food. This allows us to select the bacteria with the greatest ability to tolerate formaldehyde, allowing us to geographically segregate bacteria by this ability. In the next few decades, scientists aim to gain a quantitative and mathematical understanding of adaptation to complement the qualitative and anatomical understanding which we already possess. R is the new way to study evolution, to see it in action.